The next participant is Amit Pal.
Good evening, everyone. Today, I'm going to speak about CCRA inhibitors could act synergistically with tetracycline to prevent the spread of SEC-MSC and hence eliminate MRSA. The mobile genetic element, as we know, which carries resistant genes, is widespread among the microbial biosphere. And so we need to focus in order to stop that. And SEC-MSC, which is the staphylococcal cassette chromosome MSC, is the mobile genetic element of the MRSA. So the MSC encodes for MEC, which further codes for the beta-lactam drug resistance, and the CCR, which codes for the mobility of SEC-MSC. And this is the particular gene island which I'm talking about. So here in the schematic, we see that upon the uptake of this SEC-MSC, how the sensitive strain gets converted into the MRSA and vice versa. So when the antibiotic is in the corner, we need to, the MRSA actually excises this particular element and suicides for the community, but make sure that this element is in the pool. So we need to focus our energy to target and to stop the transfer. So the targeting the transfer is very crucial. So my hypothesis is inhibition of the CCRA, which is the mobile element, will stop the transfer of SEC-MSC, and hence the MRSA population will be under control and can be eliminated by lower doses of antibiotics. So in order to meet my, in order to validate my hypothesis, here are some objectives. First of all, we need to find out the inhibitors of CCR and test these inhibitors for the prevention of the dissemination of this particular element, and to find out the synergistic effect of a specific antibiotic and CCR in vitro and in vivo as well. So in order to find out the inhibitors of CCRA, what I did was I performed a blast search and I saw CCRA has homology with REC-A of various species of bacteria ranging from 78 to 99%. So, and as you know, REC-A is an ATPase-driven recombinase, and it helps in DNA repair, and it is very well discovered, and there are many known targets to it, inhibitors to it. So I suggested that why can't we use these inhibitors to inhibit CCRA? So the inhibitors of REC-A could be used to inhibit CCRA, and in order to test it, in order to test these inhibitors for the preventing the transfer of SEC-MSC, we could do a genomic level excision study of SEC-MSC by doing a PCR analysis of the genomic DNA of both the susceptible and the resistant strain, and go for transcriptional and protein level expression of CCR gene by Western blot, gene reporter assay, and RT-PCR. And in order to like validate my hypothesis that if we could control the transfer of this particular resistant gene element, we could check the population of the MRSA, and further, when the population will be under control, we could use a specific antibiotic in order to eliminate MRSA. So I was going through lots of papers, and I finally found out tetracycline, which is susceptible to MRSA, but in a long exposure and long dosage. So if we could, so I suggested further that if CCRA inhibition could keep the population of MRSA in control, so a lower doses of tetracycline would be enough to eliminate. So in order to prove that, we could do a simple Kirby-Boyer disk test using cefoxytin disk, where we will be using low doses of tetracycline along with the CCRA inhibitors and check the results. Further, in order to prove this same thing in vivo model, we could use cotton rat, where we could instill MSSA along with a low inoculum of MRSA and introduce our CCA inhibitors and a low doses of tetracycline into the nares. And finally, over a long period of time, we can surgically remove the nose and enumerate the type of bacteria. So the possible outcomes would be prevention of the dissemination of this particular element, as the mobility will be reduced upon inhibition of the CCRA, which helps in the mobility, and reduction in the expression of CCRA, and the MSSA mixed with the low inoculum of MRSA along with the CCRA inhibitors and tetracycline would show susceptibility to cefoxytin disk. And when we enumerate the bacteria from the nose of the rat, we could see there are possibly no MRSA in it. So, and in order to suggest an alternative strategy, as we know, REC-A is an ATPase dependent recombinase. And REC-A has homology with CCR. We could use a wide range of bacterial ATPase inhibitors in order to inhibit CCR and prove the same. So, thank you.
Nice presentation. Just so that you know from, I mean, this could be good science and always fun to study synergy or look for possible synergies. Always good exercise. But if you're looking to take this forward as an idea, you should be aware that, you know, MRSA is already a crowded area. It's an important area, but it's a crowded area. The pipelines are very healthy. Where the pipelines are not healthy is gram negatives. So if a similar hypothesis could be used to take an existing antibiotic and make it effective against Klebsiella or Pseudomonas, I think so. From a purely industry perspective, it'll be far more interesting. I'm sure this work is interesting, but from a practicality perspective, I would encourage you to look at gram negatives.
Thanks. That's a good suggestion. Thank you.
I was wondering why you selected tetracycline. Is it a general phenomenon or specific to tetracycline? Because tetracycline doesn't really pose the problems that some of the other antibiotics do in terms of toxicity, in terms of the need to reduce the dosage.
Yeah.
There's a new concept of what you call bioenhancers.
Yes, yes.
I don't know whether you are aware of them, including natural products like piperine. By adding it, you are able to reduce the dose of the antibiotic to levels which are less toxic than the normal dose. So your selection of tetracycline, I don't know, is it specific only for tetracycline or do you need to repeat it with other antibiotics?
Actually, my thing was to stop the mobile genetic transfer in order to keep the population of the MRSA to a particular extent and not increase it. And I was looking for antibiotics which could be susceptible to MRSA itself. So I was being very specific. So in that perspective, I found tetracycline, which is used in a with a longer exposure and higher dosage. So it's only because of the fact that I didn't find any other literature based on MRSA, which is required, which is possible to, which is susceptible. I mean.
So, would you envision that this target the patient who is currently colonized with the organism or future patients by reducing the spread of the mobile element within a hospital population or a regional population? Is it mainly used to benefit an individual with an acute infection or a public health issue?
Yeah, I mean, it could be used as a future perspective because if this already colonized, the spread of this element would be already there. So when suppose if a patient comes up with a fresh infection and so we could diagnose and stop the spread first and then use the antibiotics. So
Yes, I have one question. So how could you make sure that CCR inhibitor will not create any problem to the host? Because CCR is single-stranded DNA binding protein, and a lot of single-stranded DNA binding protein already present in the host. If you can inhibit this single-stranded DNA protein of the host, then it will create a toxicity. So what is the best way to rule out that it's not going to create any toxicity if you use CCR inhibitor?
yeah, the inhibitors which I went through contained curcumin, so and other natural compounds, so that wouldn't show much effect on the host itself. I mean, curcumin is obviously we have it and the turmeric, so it's widely accepted in India.
But the curcumin, if you put in cook, most of the function is not at all over. So once you cook, the curcumin function is almost gone.
Yeah, like we have this habit of having curcumin with tulsi in the morning every day.
Okay,
So
then it will work definitely. Okay, thank you.
The next participant is Pyare Mohan Das.
First of all, good evening, everyone. I'm Pyare Mohan, a biotechnology undergrad at Amrita School of Biotechnology, here in front of you on stage to share a few of my thoughts on bacterial persistence. bacterial persistence is a major cause of antibiotic treatment failure by relapsing infection. It is increasing the clinical burden of in treating chronic infection, and it has also been found to be acting as a catalyst for the emergence of genetic resistance. Now, if you want to look at what happens in the phase of bacterial persistence is, this specialized cell avoid antibiotic killing by entering to a physiologically dormant state that is at the cost of complete retraction of its growth mechanism. Now, if you can see in the diagram, a regular cell, when interacts with few factors, which activates the mechanism which induces the formation of persister cells. Now, now there is a big debate going on whether the persister cells can be activated stochastically, but we won't go to that now, as obvious reasons as the stress reagents, which includes sublethal antibiotics, and also physical agents like temperature, pH, which randomly activates the mechanism which induces the formation of persister cells, to which the well-known type of antibiotics fail to target because all the machineries they usually target to remains inactive in case of persister cells. Now, following to the characteristic of persister cells, there is a well-known mechanism in which an initial bacterial population is treated with a lethal dose of antibiotic, which gives a biphasic kinetics in which in the initial blue phase has a rapid drop in bacterial count, which represents the majority of killing of bacterial population, followed by a second phase of much slower kinetics, which represents the poor killing of persister cells. Now, it is currently unknown why the tolerant persisters are getting still slowly killed in the second phase. And the second characteristic is these persister cells, after withdrawal of the antibiotic, are capable of reproducing the initial bacterial population, which includes the antibiotic sensitive cells too. Now, the second factor of interest to bacterial persistence is the SOS response, which is among the cell signaling mechanism to the upstream of bacterial persistence. Now, it has bifold function. First function, it modulates the persister formation when gets induced by a stress response, and it helps in the resuscitation of persister cells by providing diverse DNA repair mechanism. Here comes REC-A. When gets activated, it induces the stress response or the SOS mechanism, the DNA repair machinery in bacteria, which again induces the formation of persister cells. Now, if you go back to the previous experiment, if we check the regulation of REC-A in this whole mechanism in a time series manner, the REC-A should be, the concentration of REC-A should be similar in the initial phase because no DNA damaging agent has been treated to it. But when in case of persister cells, it should be overexpressed or it should be highly regulated. This is how it is keeping itself safe from DNA damage. But a few cases has been found out that overexpression of REC-A actually causes resistance. Now, following up to all these observations, an assumption has been made that persister cells have preferential activation of DNA repair mechanism by regulating REC-A. And in order to do that, we have to study the regulation of REC-A in persister cells with stress and without stress, or more likely after withdrawal of stress condition. Now, in order to perform that, we first label the bacterial cells with the help of genomic targeting by GFP, and if we may, we'll be able to use the CTX5 vector, which has been shown to irreversibly attach the GFP to the bacterial genome without hampering any known open reading frame, followed by a flow cytometry study of the biphasic killing using well-known viable dyes like PI, that is propionium iodide, or also CTO9. And then we compare the regulation of REC-A in sensitive cells and persister cells and even resistant cells. And the possible outcomes will help in establish the dynamic regulation of REC-A in both conditions, that is stress and then after withdrawal stress condition, followed by, and we can also establish the new insights on the cellular decision-making mechanism, more likely in the SOS response, which is upstream of positor formation. And before going to the future perspective, I'd like to wrap up a little bit. Given that endurance is the key property of persistent cells, there is no doubt that is hampering our effort in treating chronic infection, and a new approach to antibiotic killing and the timing to antibiotic treatment, which guarantees the complete eradication of the pathogen, will certainly help in the effective use of bacterial therapeutics. Thank you.
So there are probably a lot of different mechanisms for ultimate bacterial persistence. They can be very slow growers. They can hide out inside a cell. And maybe they can induce, high level of tolerance in the host, so allows persistence. Is there a particular mechanism of tolerance that might be the most susceptible to your drug therapy and one that might be more difficult in your mind?
Sir, regarding the cellular signaling mechanism upstream, or which induces the positor formation, there are actually a lot of mechanisms, but if you go for the study, there has not been any congruence within the studies. So it's still in the phase of infancy, and the reason why I have chosen SOS response, because it has bifold function. It helps in inducing persistence, and it also helps when the persister cells are going again reproducing the antibiotic sensitive cells. That's why.
And what do you think is the strongest inducer of the SOS response while the organism is in the host? Why are they undergoing that? I know, why is the bacteria experiencing the SOS response in the host? Is it in response to the immune system, in response to treatment, in response to other bacteria?
Sir, the SOS response in bacteria is actually the DNA repair mechanism. So, sir, whenever there will be a DNA damaging agent that enters the bacteria, then it will start damaging the DNA, and more importantly, when the single stranded DNA will be formed, Rec A is the protein that gets activated, binds to the single stranded DNA, and then induces the SOS response. The damage will certainly happen if any DNA damaging agent, that could be that could come from any pathogen that enters the body. The stress agents includes sublethal antibiotics sometime, and even physical stress agents like temperature, pH. They sometimes also hampers the damage to DNA.
Quick question, is it only Rec A in DNA repair, or are other DNA repair enzymes important?
I haven't looked at all the mechanisms which are responsible for activation of SOS response, but there has been studies in which they have looked at more importantly, they have said that Rec A to be the first or sole inducer or the initial inducer of SOS response. Well then, thank you everyone. Have a nice day.
Yes.
Sorry.
I have one question. So when you are using some antibiotic, if that antibiotic creates some problem to the DNA, that time SOS response will be activated. But if you use like beta-lactam antibiotic, that has nothing to do with the DNA repair system. So how come that induce this SOS response in bacteria?
Sir, so there is no doubt that beta-lactams or aminoglycosides or fluoroquinones, all these enzymes do not are unable to target positive cells, but in that case SOS response might not be really getting activated, but the whole point is the activation of persister cells. So I'm looking at SOS response, probably I'll be studying with the antibiotics that will be damaging the DNA.
But most of the 70 to 80% of the antibiotic we consume that do not target the DNA. So most of the antibiotics target the beta-lactams we are using.
Sir, but then again, those antibiotics, when they enter the pathogen, not directly damage the DNA, but they might end up damaging the DNA. And once the DNA gets damaged, there is no possible reason to the bacteria to produce itself and expand.
Okay, thank you.
Thank you, sir.
The next participant is Akshaya Balasubramanian. Good evening, ladies and gentlemen. I'm Akshaya Balasubramanian, a third-year undergraduate of ASBT. So my hypothesis actually centers around a two-component signaling system in Staphylococcus aureus. Now, Staph aureus is a human pathogen, and it's one of the leading causes of nosocomial disorders such as endocarditis and osteomyelitis. And unfortunately, due to antibiotic resistance, there are limited treatment options. So looking at this organism's pathogenicity and also taking a look at new drug targets has become especially important in today's scenario. So with keeping that in mind, I'm looking specifically at the SAERS two-component signaling system, which is a key regulatory system of exoprotein virulence factors. So this is a diagrammatic representation of the system. Over here, if you'll notice, this is SAES, which is an intramembrane histidine kinase. So it senses external cues and stimuli, autophosphorylates, then transphosphorylates SAER, which then binds to DNA and causes transcription and consequent expression of the virulence factors. However, as you can see, there are absolutely no extracytoplasmic domains by which an environmental cue can be sensed. So that brings us to the question, how exactly is this happening? So what we do know, however, is that there's a transcription factor called FER, which is iron uptake regulator, and that has been shown to have to be a positive regulant of SAERS. But FER happens to be a repressor, and for a repressor you need to have a binding site, which is lacking in SAE target genes. So obviously the relation between the two has to be indirect and through an accessory protein. So other two-component systems, such as in Bacillus subtilis as well as in Staph aureus, have showed that many of these environmental sensing proteins, accessory proteins, are actually ABC transporters, which brings me to my essential hypothesis that perhaps the accessory protein of the SAERS TCS system is a FER-regulated ABC transporter. I've specifically identified ISDEF and HTS-ABC, not because they're only FER-regulated, but they've also both been shown to have an effect on virulence, specifically those that SAERS regulates. So just to give you an idea of things in a very simplified schematic, here you have your cytoplasmic iron that essentially binds to your transcription factor FER, which increases its affinity towards to its respective DNA binding site, which is a FER box, which is present upstream to the ISDEF and HTS-ABC genes. This in turn ensures that ISDEF and HTS-ABC are not transcribed, and hence the ABC transporters are not produced. Now, my hypothesis is coming actually in this region where I'm essentially interconnecting the fact that perhaps it's preventing the auto-induced SAE transcription, which in turn results in decreased levels of phosphorylated SAER, which decreases the expression of the SAER's target genes. I'd like to study this essentially by looking at the effect of ISDEF and HTA-ABC mutants on SAE transcription levels using Northern blot and qRT-PCR, and with primers and probes designed against SAEP and SAER, which are products of the SAE operon. And I'd also like to look at the expression profiles of the SAER's target genes using 2D Degen mass spec. Also, I'd like to just, this is a little offshoot actually, but to check the effect of exogenously added iron and Heyman on the mutant strain SAE transcription and the target gene expression, because essentially the ion that is present that binds to your TF transcription factor FER has to come from somewhere. So which is from heme or iron that's present in the neutrophil granules. So if my hypothesis is correct, these would be my expected outcomes. So there will be a decrease in SAE transcription levels in my mutant strains, as well as decreased gene expression. And there should also be the repression that we see in the presence of iron and Heyman should also essentially be nullified. And if all of this happens, hopefully I'll be able to identify either of these or both ABC transporters as accessory proteins to the SAERS TCS system, and perhaps take a look at it as a possible drug target. Thank you.
So, you know, if you advanced all the way to the last stage and were envisioning them as drug targets, how do you think they would be working because of their effect on the target virulence genes, you know, downstream of this two-component system, or could they simply be working at the level of iron as a cofactor for the organism, or both, I guess?
I actually think both, personally, because as ISDEF, specifically as an ABC transporter, is extremely important when it comes to iron uptake. HTS ABC with respect to heme. So yes, there is a possibility that we'd actually end up affecting the organism more as a result of lack of iron uptake rather than of the virulence factors. But this is just a question, a basic research question that just interested me, quite frankly. The future perspective, I'm still a little shaky on. I need to do a little more reading to be able to confidently say that this is going to be it.
I believe there has been some research on the ISD system, or at least a surface exposed component as a vaccine antigen that might also inhibit virulence. I can't remember how that trial developed, but it's my it's not really a question. I just recall that as a drug target, but maybe you've heard something along those lines.
Yes, I have, but I unfortunately didn't get to the level of that reading yet. I have done maybe 2/5 of the reading that I'd be happy doing and officially presenting this as a grant proposal. Thank you.
You had a question, sorry.
Yes, sir.
Oh, please, I didn't see it. Sorry.
So you are going to target one essential protein for your drugs. But you know, if you target an essential protein, all the time cells will survive, will try to survive, and they will make resistance. So how could you convince that pharma company this is the best target and your resistance will not go quickly?
So essentially a resistance mechanism develops around something that ABC transporters are absolutely essential. They're highly conserved. The chances of them changing their structure or in any way as to make them resistant in themselves as a structure is very low. if there is a resistance mechanism, perhaps it will evolve around by, I don't know, nullifying my drug like an enzyme or something like that. But even if that is the case, you can perhaps look at natural products or some such that would block the ABC transporter that wouldn't have a resistance develop towards it. This is just off the top of my head, so forgive me if I haven't answered it to your satisfaction.
Okay, thank you.
Thank you.
The next participant is Shrisha Sridharan.
Good evening, everyone. Myself, Shrisha Sridharan. So my hypothesis is revolving around one of the most common infectious disease that has afflicted worldwide, and that is nothing but tuberculosis. So tuberculosis, as you all know, is an infectious disease which primarily affects the lungs, and it is caused by the pathogen Mycobacterium tuberculosis. So this particular schematic representation actually illustrates the common pathway taken up by the Mycobacterium tuberculosis once it infects the host. And research has focused on this particular area, as well as many of the virulence factors of Mycobacterium tuberculosis in order to design drugs and other novel therapeutics. However, with the emergence of extensively drug-resistant and multiple drug-resistant forms of tuberculosis, there has been a constant demand to look into different aspects which can be possible targets of novel therapeutics in the future. So this particular slide basically shows gives an idea of the global statistics of tuberculosis. Now, coming into my hypothesis, this is a particular paper which was published recently in Cell. And this particular paper actually talks about an alternate pathway which exists via which the Mycobacterium tuberculosis can cause infection, where the cervical lymph nodes are primarily infected. And the pathway is via the M cells or your microfold cells. Microfold cells are nothing but they are a specialized type of epithelial cells which actually are present in the mucosa-associated lymphoid tissues, whose primary goal is nothing but to deliver the antigens to produce mucosal immunity. So what is already known on this particular aspect of M cells is actually the pathogenesis of E. coli and Salmonella, and of course Brucella abortus, which is a zoonotic pathogen. So the research and the recent findings have said that in the case of E. coli, Salmonella, and other gram-negative pathogens, they enter the M cells via the glycoprotein 2 receptor, which is a well-characterized receptor of the M cell. And in the case of Brucella abortus, it is the cellular prion protein receptor via which the bacteria enters. And the bacteria expresses HSP60, which helps in the process. So this brings me to my hypothesis. What will be those factors of Mycobacterium tuberculosis which enables the bacteria to get inside the M cell? So if we can target these particular molecules or proteins, we might end up in a novel therapeutic. So it's always better to start with what we already know. What I propose is that in the case of E. coli and Salmonella, the interaction happening with the glycoprotein receptor is because of the expression of a FIMH protein which is present in the type 1 fimbriae. In the case of Mycobacterium tuberculosis, there is a type 1 pili. So we can actually go for an in vitro binding assay where we can use mutants of Mycobacterium tuberculosis where they are mutated for the adhesion-causing genes. And then we can carry it out by doing an immunostaining where we can look into the interaction between the proteins. If there is a successful internalization, we can again do an affinity chromatography to further find out the protein of interest. There we might get the novel target. And then we can, if it is successful, we can go for an in vivo study. So this actually applies to not just the glycoprotein receptor and the protein involved in the internalization by interacting with the glycoprotein receptor, but also to the cellular prion protein, because Mycobacterium tuberculosis also expresses HSP60. And one more important factor is that these two are virulence factors. So as Dr. Nizam mentioned in the morning, if we can actually target the virulence factors, then who knows, we may not have drug-resistant forms of tuberculosis in the future. This is one approach, but if I do not get possible targets which actually helps in the entry of the pathogen into the M cells, then I wish to go for a broader study where I can do a blast and find a blast or molecular docking studies so that I can find out of all the proteins which are expressed in Mycobacterium tuberculosis as well as the receptors. And I can find out the regions of similarity. And then I can go for an RNA interference study. And then I can go for a pull-down assay. So that is a specific protein-protein interaction study. So I can narrow down my targets. And then I can again go for affinity chromatography. And again, if I get a novel target, I can go for in vivo models. So the expected outcome is that by doing all these studies, I might be able to find out those molecules which are responsible for the entry of Mycobacterium tuberculosis into the M cells. And thereby, we can target this particular pathway so that the entry is prevented. And if the entry is prevented, the dissemination or the manifestation of the disease can also be prevented. Thank you and have a wonderful day ahead.
So I was thinking about the HIV individuals, the rare cases that they don't get HIV because their receptors mutated. Could you, as well as looking at the tuberculosis pathway, could you actually just target those receptors directly? What was it, the prion receptor and GP2, or is there going to be a problem targeting those receptors?
So I'm not very sure whether these receptors are present in that case also, but in the case of M cells, these two receptors are well characterized. So yes, I believe that in the case of HIV-infected patients too, these receptors can be targeted.
Sorry, not those receptors in HIV, it's just the idea of the receptor being wrong stops the infection. I was just wondering if, in this case, if you could target these separate receptors and cause resistance to tuberculosis, or is it that the receptors, if you do something to the receptors, that's actually going to be bad for the M cell?
I actually focus on mutating the proteins which are responsible in Mycobacterium tuberculosis rather than mutating the proteins on the M cells. So I don't think so that is going to be.
I was interested in your schematic, which had the alternate strategy for candidate identification there. So does the RNAi study inform the pull-down assay, or isn't a pull-down assay an unbiased approach that is its own independent starting point? Or how does the RNAi data feed into the pull-down assay? You're looking for which host protein will be the bait in a pull-down assay? I'm trying to understand that connection between those two steps in the pathway.
So what I thought is that using an RNAi study, while doing the blast study, you might get a large number of results of possible interaction between the receptors and the Mycobacterium tuberculosis. So to screen it down, as a first step, you can go for RNAi studies and that has been done in other cases. And again, you can narrow down the list. So in pull-down assay, the advantage of a pull-down assay is that you get a specific protein-protein interaction. So there, you can, if you get a specific protein-protein interaction, that can be carried out, for instance, by mass spectrometry, and then you can analyze that. So that was my idea behind.
I think I understand you. So the RNAi would identify a host protein, a M cell protein that is required for the mycobacterial entry, and then you would use that protein to try to pull down mycobacterial factors that are the ligand for that protein, or is the RNAi being done on the mycobacteria?
It's the RNA done on the mycobacteria.
Ah, okay.
It's the other way around.
And that's an efficient procedure for mycobacteria? Maybe, I don't know.
I'm not very sure of it.
Yeah? Okay.
Your title says a novel way of combating tuberculosis. That's a very, very tall order. Now, what is the current state of knowledge in this particular area related to your work?
So what they have found out till now is just that MTB can be internalized by M cells. That's it. And from there, we'll have to actually start from scratch. That's what I mentioned in my presentation, that only if you can target, only if you initially can identify the molecular mechanism of entry can you go further and beyond. So that was my idea behind putting a novel idea. Title.
It needs a lot of multidisciplinary efforts.
Yes,
right?
yes.
And which part of it is your strength or you want to concentrate on?
I would like to start with what is already known in the case of E. coli and Salmonella, because since that is already known, it would make my work easier.
So you extend the current concepts on E. coli and Salmonella to
Mycobacterium tuberculosis.
Is it a reasonable logic?
There too, it is just a hypothesis. I believe that since FMH in the case of E. coli, if I take E. coli, since FMH protein is involved in the entry, and since type 1 pili in Mycobacterium tuberculosis is involved in addition to epithelial cells, there might be some components. I believe that there might be some components which can be characterized. That is the first case. And the second case is the interaction between HSP60 and the PRPC receptor protein.
Because if it works, it's a fantastic thing. The only thing is, what is your confidence level that this current knowledge can be extended to tuberculosis scenario or not?
I'm confident about it.
Yeah, very
good. Good answer.
Thank you.
If you want to do this work, right, beyond the hypothesis, if you get into doing work, can you actually do work here in Amrita? Because I think you need BSL 3, if I'm not mistaken.
I'm not very sure of the technical aspects, but
something to think about.
Even if I can't do it out here, I'm sure that all my faculty members will help me in reaching somewhere to do the work.
In fact, I tell you, what he mentioned is extremely relevant.
Sorry, sir?
Because in our, what he said is extremely relevant.
Yeah, I know.
The kind of containment facilities that you really
We
wanted,
required.
very few centers in India have. Forget about Amrita.
Yeah, I know.
Because you need compartments where the experiments have to be done naturally with glove compartments.
I realize.
And then the treated in vivo models have to be sucked into an incinerator and burnt. It cannot come back to the lab. It goes beyond even BSL 2 or 3. That's what I'm saying.
Okay.
And at the moment, I don't know in India we have any facilities to do this. Where CDRI certainly doesn't have, and that's supposed to be the most sophisticated.
Astra is closed.
Astra is closed.
Astra is closed. IIT Mumbai, I don't know.
I don't think so.
Something to think about, definitely, because there could be practical limitations.
Anyway, if you reach
Yeah?
that level, you already achieved something.
Absolutely.
Thank you, sir. Thank you so much, sir.
The last participant is Natasha Menon.
Namaskar and good evening. My name is Natasha Menon, and today I'm going to be talking to you all about clove bud oil. For ages, clove bud oil has been used in traditional Indian medicine for various ailments and illnesses, but today I'm specifically going to be talking about clove bud oil as a potential therapeutic that can attenuate bacterial virulence, specifically Pseudomonas aeruginosa virulence, and enhance host immunity. So our story starts with Pseudomonas aeruginosa, which is a gram-negative opportunistic bacteria, which is the cause of a large percentage of nosocomial infections. What's interesting is a large number of the virulence factors of Pseudomonas aeruginosa are actually regulated by a system known as quorum sensing, which is a microbial communication system. Now, actually, through research that's been done here at Amrita School of Biotechnology, it's been found out that clove bud oil actually attenuates a wide array of these virulence factors through interfering with quorum sensing. So if you look at this figure here, as you increase the concentration of clove bud oil, you're seeing an increase in the inhibition of biofilm formation, as well as an increase in dispersion of pre-existing biofilms. So it's interesting because biofilm formation is, it enhances virulence in Pseudomonas aeruginosa, and it's also under the control of quorum sensing. Lastly, many of these quorum sensing signal molecules, or QSSMs, have been found to have an effect, an immunomodulatory effect on the host immune response. So since we know that quorum sensing enhances bacterial or Pseudomonas aeruginosa virulence, and it also is able to modulate the host immune response. And we also know that clove bud oil can inhibit bacterial virulence. It should be that clove bud oil also can modulate the P. aeruginosa mediated host immune response. So why is this work significant? I think today many people have discussed the fact that we're dealing with an antibiotic resistance crisis. And so when you target virulence factors instead of trying to aim to kill bacteria, you're actually less likely to result in resistance. So, and an additional advantage with clove bud oil is that you're having this immunomodulatory effect. So a lot of bacteria have the ability to either evade or suppress the immune response. So if clove bud oil is able to have a two-pronged approach, that's always optimum. So this flowchart kind of depicts the methodology that could be taken to address this issue or to figure out how clove bud oil might have an effect on the host immune response. So there's 4 aspects to this, actually. So here you have your effect on neutrophils, effect on cytokine production, effect on T lymphocytes, and then lastly, effect on the whole system through in vivo studies. So if you're looking at neutrophils, the preliminary test would be to look at neutrophil activation or neutrophil killing, or both. And if clove bud oil enhances this, then it would be interesting then, and this would actually, you would look into this by looking at aureus production, reactive oxygen species production. And then it would be interesting to see how the neutrophils are able to kill the pathogen. So there are 3 methods of killing. That's either through phagocytosis, degranulation, or netosis. So, and I've listed down how you could do that. I'm not going to go into detail because of time constraints. But secondly, cytokine production. If we could see how neutrophils, when you treat with clove bud oil, how the neutrophils respond in their cytokine production. So that could be either pro-inflammatory or anti-inflammatory cytokine production. That could be easily done through like ELISA or Doppler assay. Thirdly, looking at the effect on T lymphocytes, that could be done through a co-culture system. And that's important because that's looking at the adaptive immune response as well. And lastly, after looking at all of these different aspects, it'd be interesting to look at in vivo studies, maybe through a mouse model. So the expected result is that clove bud oil should have some sort of immunomodulatory effect in the host response to Pseudomonas aeruginosa infection. And therefore, this agent, clove bud oil, could be a potential therapeutic. And thank you.
So if you hypothesize that a key aspect of the action would be to interrupt quorum sensing and biofilm formation, and then you had exciting results in your studies with the immune cells, which type of infection in the animal do you think would be a good starting point to study potential therapeutic effect, one in which a biofilm might be an important component?
Biofilm formation, I mean, essentially, it enhances virulence. So it could be maybe through, I know for certain lung infections with Pseudomonas aeruginosa, you have a lot of biofilm formation, which then causes a lot of different effects with the immunomodulatory response as well. So that could be one aspect. I'm not exactly sure how to go about introducing a lung infection in a mouse, but that would be one aspect.
You put the mouse to sleep to start with. Then I guess another question is, so which virulence factors are being made by the Pseudomonas that provoke changes in host immunity? Are there specific examples or leads that you're thinking about, or is it more just kind of a high concept that these interactions are likely to occur?
I mean, quorum sensing and Pseudomonas aeruginosa, there's a lot of different virulence factors that you're looking at. You have elastase A and B, you have piocyanin, we have pyoverdin. And a lot of these do have an effect on the host immune response as well. I believe pyoverdin or piocyanin, one of them has an effect directly on causing some sort of toxicity in the system with effect on immune cells. So.
Clove bud oil, I think it kind of burns if you put a lot on your skin and stuff. And I was just curious what sort of dosages you would use to try and look for an effect.
Well, I think if you're looking at if you're looking at a therapeutic aspect of it, there would probably have to be more characterization involved. I think you couldn't just clove bud oil. Maybe you'd have to look at exactly, like I know one of the major components of clove bud oil is eugenol, which I think there's been a lot of studies on that as well. So looking exactly at how that affects the system, and if you apply that, I'm not sure if you would have that same burning sensation. You might, but it would be one thing to look at also, is exactly what causes this kind of effect in the clove bud oil.
I have one suggestion and one question. Suggestion is, even if you don't look at this as a therapeutic, you can look at it as a biofilm paint. If it works properly, I mean that the pathways are much easier than doing a therapeutic, right? Obviously. So the industry is looking for substances that can prevent biofilm formation on devices, on various surfaces, right? For home use, for hospital use, etc. So if clove oil can actually prevent and or disrupt biofilms,
Yes,
something
sir.
to look at as an application, because the pathway is so much easier than the other stuff. Question for you is, which is a common question I have with all these natural products, is while they're fantastic, how are you going to go about figuring out which is the active component? Because they're all polypharmacology, right? And inside the clove, they're happening in particular combinations to make a particular effect happen. 2, 3, 4 of them. Have you had some thoughts about how you're going to figure out that one or two components that are causing that effect?
So first of all, it would be important to know what is in clove bud oil. So as I said earlier, one of the major components is eugenol. So if you wanted to see, maybe find out, is eugenol the component that is causing this action, maybe you would have to somehow get eugenol, just purified eugenol, see if eugenol has a similar effect when compared to clove bud oil. And that way you can kind of make some sort of correlation or assumption that clove bud oil is the active component in clove bud oil. Eugenol is the active component in clove bud oil, sorry.
You're actually claiming two biological activities. One is bactericidal or bacteriostatic activity, and there is immunopotentiation. We don't know the relative components of these two biological activities, number one. Of course, I was also going to talk about eugenol, because eugenol is a major component. And I'm sure that there are MIC figures available on eugenol. Have you looked at them? MIC figures available on eugenol?
Yes. So actually, through the work that was done here, we actually also did look at eugenol. So not only just clove bud oil was looked at, eugenol was also looked at, and seeing how the effect was with that. So it's just I don't think they did an extensive study with the eugenol, but.
Because as he rightly said, if the active component is eugenol, which I don't believe it is, perhaps in most of the natural products what happens is the activity does not reside in one component. There's a lot of synergistic effect even with micro components of other things, where the activity of the oil itself may be much more than the additive activities of single ingredients. That is to be looked into. Second thing is that, I was telling Dr. Banerjee, without knowing that your hypothesis here, I was reading the autobiography of Dr. P.K. Warrior, who is a doyen and patron of Ayurveda in the whole country or the whole world. Now, he was talking in terms of treating one of his patients who was a chronic smoker. He was smoking 40 to 50 cigarettes a day. So he came to him, he wanted to wean himself off of smoking. And he prescribed clove bud oil. And he said that within 3 weeks of every day being treated with this, he was 100% weaned off from smoking. He had no interest in smoking anymore. So I told the cortical people, if that is really true, today, you know, there are a couple of products based on nicotine, which are smoking cessation products. One is Nicotex, another is Nicoderm, a transdermal delivery system. And they together are $1 billion products. If our simple clove oil can do the same thing, I don't know what's preventing us making a soft gelatin capsule of clove oil and giving it as a drug. I mean, this is the kind of thing we should be thinking in terms of.
Yeah, maybe that could be the next step, is looking at the effect on clove oil on nicotine.
And also, you know, it's being exported in large quantities by a company called Plant Lipids in Cochin. I still do not know what they use it for in the American and the European market, because they're exporting to developed countries. It'll be interesting to find out what they are using it for. I don't think not as a bacteriostatic. I doubt it. Because when you talk of bacteriostatic, bactericidal, there are so many synthetic things available. I don't think anyone will resort to natural products. It'll be worthwhile looking at that.
Thank you, sir.
Yeah. Thank you.
Thank you.